In this video, we will discuss about the concept of immunity and immune response. What is immunity? So it's a state of having sufficient biological defenses to avoid infection, disease, or other unwanted biological invasion in our body. Being immune to a specific disease means that our body has the ability to recognize and reject the agent such as bacteria, viruses, or pathogen that causes that particular disease, hence prevent infection. The biological defenses is provided by our immune system. So it is the immune system that gives our body the state of immunity, when it is able to recognize and respond towards any unwanted biological invasion that is potentially harmful to our body. There are two ways the immune system will respond towards any entry of unwanted molecules: the innate immune response or the adaptive immune response. Innate immune response, also known as the non-specific immune response, is characterized by its ability to rapidly or immediately respond toward biological invasion. The innate immune response is part of the first line and the second line of defense of the immune system. The two categories of the first line of defense is the physical barrier and the chemical barrier. The physical barrier includes our skin, formation of urine, and hair. For the chemical barrier includes saliva and gastric juice. If the foreign molecules or the pathogens was able to breach the first line of defense, it will come into contact with the second line of defense of the immune systems that consists of phagocytes, antimicrobial proteins, as well as the inflammatory responses such as swelling and redness. Essentially, the innate immune response provided by the first line and the second lines of defense of the immune system function to create barriers to keep foreign molecules out of our body or to limit their ability to spread throughout our body. This type of immune response is always general or non-specific. Why? Because it will respond towards anything that is considered as foreign to the body. And the mechanisms of response is the same for all different types of biological invasion. In other words, the innate immune response for bacteria is the same as the innate immune response against viruses. If the biological invasion was able to evade both the first line of defense and the second lines of defense of the innate immune response, only then the adaptive immune response will come into play. The adaptive immune response, or also known as the specific immune response, consists of humoral immune response and cell-mediated immune response. It is part of the third line of defense of the immune system. These types of immune response takes longer time to respond towards the biological invasion, hence the slower response time in comparison to the innate immune response. The adaptive immune response is highly specific. It has the ability to differentiate even the smallest differences between the two different biological invasions. More importantly, the adaptive immune response has the ability to remember all the specific types of biological invasion that it has previously encountered and protects our body against the same biological invasion in the future. That was the types of immune response. Next, we will look at antibody and antigen. Antibody is a type of protein or immunoglobulins produced by the plasma cell in response towards specific antigen. Antibody have the ability to react against the antigen. What is an antigen? Antigen can be any foreign substances capable of stimulating the immune response. Antigen are made up of protein or large carbohydrate. Many antigens protrude from the surface of the foreign molecules, means that it sticks out on the surface. For example, here we have the virus for COVID-19. As we can see in the diagram, on the surface of the virus for COVID-19, we can see there are three different types of proteins sticking out. These are the spike protein, used by the virus to penetrate into the host cell and causes infection. Our body immune system recognizes this protein as foreign, means that it knows that this protein does not belong to our body. So, if our body immune system encounter the spike protein of the coronavirus, it will initiate immune response against the protein. So, since the spike protein triggers initiation of immune response, this is why the spike protein is called an antigen. Other antigen includes toxin produced by bacteria, for example, cholera toxin. This is the toxin produced by Vibrio cholera, the bacteria, which causes food poisoning. Next, all antigen have specific site called epitope, where the antigen binding site of the antibody or the paratope can recognize and binds to. Different antigen will have different types of epitope, which will allow the body immune system to recognize and differentiate the different types of antigen, and serve as the basis for the specificity in the adaptive immune response. Next, we will look at the structure of antibody. All antibody is made up of a Y-shaped glycoprotein, and the glycoprotein forms bivalent, bonded together by the disulfide bond. Each of these bivalent act as one unit of monomer for the antibody. There are 4 polypeptide chains that make up for the antibody, 2 heavy chains and 2 light chains. The heavy chains have a larger size compared to the light chains. The bivalent previously mentioned refers to association of the heavy chain and the light chain by disulfide bonds. Next, both heavy chain and light chains contain variable region and constant region. The variable region contain identical antigen binding site or paratope that is different for different types of antibodies. The constant region is the same for all the different types of antibodies. However, other proteins associated with the constant region will differentiate the different classes of antibodies. Other than determining the classes of antibodies, the constant region is also important to act as the Fc region, which will bind with the Fc receptor found on many phagocytes, and also to bind with the antimicrobial proteins or the complement proteins. Next, there are 5 classes of antibodies based on the constant region. There are IgD, IgE, IgG, IgA, and IgM. The Ig refers to the antibody, which is immunoglobulin. IgD, IgE, and IgG exist in the form of a monomer. The IgA exists in the form of a dimer, while the IgM exists in the form of a pentamer. That's it for the structure of antigen and antibody.